Our next speaker is Marta Varela. She's going to tell us about computational models meet data learning, how physically informed neural networks can improve our understanding of the heart. Or do you want this one?
No, it's the same one.
Oh, it's the same one. Oh, I didn't see the pins. All right, thank you so much.
Thank you. Thank you so much. Thanks for the invitation to speak to you today. So I'm going to be talking to you about physics-informed neural networks and what we've been doing with them, modeling cardiac EP and the behavior of antiarrhythmic drugs, and then at the end some modifications we've implemented to address some of the problems of pains. So I think we all have similar aims here in our workshop. We want to understand physiology, understand when things go wrong and predict them, and also try and do something that corrects that, or controlling our system, which is the heart. And there are two ways of going about that. You can trust that you know how the heart works very deeply, and you can describe how it works using differential equations. You know the parameters of those equations and the initial and boundary conditions, and you have several sets of numerical tools to address this. Or you can come from the opposite end, that you have a lot of data, you're not so interested in the mechanics of things in equations, and you can create a model that learns from those. I think in real life, and for that you use neural networks. And I think in real life, we're somewhere in the middle of this, that we have some data, we have some mechanistic knowledge, and we want to combine this to create the best model we can. And there's several tools, sometimes grouped under the term physics-informed machine learning, like the techniques that Matteo was telling us about. And I'm going to talk to you about a specific type of these techniques called physics-informed neural networks, or PINs. So just very quickly trying to introduce PINs to you. If we focus on this example problem, you have some ink going into a vat of water, diffusing through a space, and you have some data that you've acquired for the concentration of ink at some time and locations in this vat. And you can create a neural network, so you can give spatial and temporal locations as the input, and then for the points for which you have data, you can enforce that there's an agreement between the predictions of your network and your measurements, and you train the network to agree with those. This is not going to go well if you have a very sparse amount of data, but the diffusion equation can come to your rescue, and you can encode that into your neural network as well. You can write it in the form of a residual form, such that if this is close to zero, then you're obeying the diffusion equation, and do that for all points of your spatial temporal domain. And you can take advantage of the fact that these spatial and temporal derivatives are what you're doing to optimize the network using automatic differentiation, and so come for free. And so this is the setup of a physics-informed neural network. Two main loss terms: agreement with the data where you have it, and agreement with the equations across the entire domain, and different weighting terms for this if you want. So this should give you a solution for this equation across all points of the domain, and it doesn't involve any discretization. It really is anywhere in the domain there. And this is the forward mode of PINs. You can also run it in what's called the inverse mode, where you do exactly the same thing, but you can also estimate some of the parameters from the equation in the same go, for example, the diffusion coefficient in this example, also using the same gradient descent that you're using to train your neural network. So what we did in the past was use this to model cardiac electrophysiology. So just very quickly, if this is not your field, we're modeling the propagation of electric signals across the heart. And we keep track of this variable V, which is the membrane potential across the membrane of cardiac cells. And you can use a simple system of equations such as this model, where you have V, the potential difference that diffuses across the heart and also evolves non-linearly, and another variable to make it refractory. And so in the examples I'm going to show you, we're going to estimate the diffusion coefficient and these other parameters that affect the properties of the action potential. So this is what we did before. This is the same PINs that I've just shown you, but I'm also incorporating explicitly the loss terms concerning the initial and boundary conditions, and now we have two variables here because we have two variables in our model. And this works very well. You can see that it gives similar solutions compared to the numerical simulations that we see here, and that we can estimate the parameters of the model that I highlighted to you, particularly well when we are in this regime of planar waves, and a little bit worse if we have spiral waves in our system. We can also do this for spatially varying tissue properties. So if your diffusion coefficient, D, is varying across the tissue, for example, here we introduced this square region with a lower diffusion coefficient as a simple way of modeling, for example, fibrosis. And you can introduce another branch to your network to estimate T everywhere in this domain, temporal domain, and it does a very good job. So here's the outcome with a map of D estimated by pins, and you can see it overlaps the ground truth square of reduced diffusion coefficient, and we can estimate the global parameters well at the same time. So we've done a few, this was what we published a few years ago, and since then we've been working on improving the setup. We are able to also simulate the activity in a spiral breakup regime. This is also with the Alev-Pamphilov model, and it also shows very good reproducibility, and also in 3D. This is a very simple 3D model. It's a spherical surface with a given thickness, and this is propagation. So you have ground truth on the left and the pins simulation on the right, and also similarly for spiral waves. And we're working on making these 3D models more realistic as well. So you could argue that, so for this is also very nice, but I've only been showing you simulated data and how the spins perform if you're working with experimental data. And if you are, then a couple of issues appear. Your model might not be a truthful representation of what the system is doing. You certainly have noise, and often you have very complicated systems of equations, more than the ones I've just shown to you. And the question I'm asking is, are pins ready for this kind of problems? And I hope to convince you they are. So I'm not going to talk about the model mismatch so much. There's work from Maxime's group in Imperial looking at this, and this is a particularly exciting development, I think, with pins with sparse regression, which is like a pins version of Cindy, if you're familiar with that algorithm, where you can learn the equations directly from the data in a parsimonious format. So before I talk to you about how to address the other issues, I just want to demonstrate an application of pins on experimental data. So the question we're trying to answer is which ionic channels do different antiarrhythmic drugs affect? And we have data from optical mapping. So this is a 2D rectangle of tissue with data in two different locations. It has a voltage-sensitive dye so that it looks bright when these cells are activated. Signals are quite noisy and messy, so they need a bit of pre-processing. And then we use the Fenton-Kalmer model to analyze these data. So here in the Fenton-Kalmer model, our previous V is now the U, and you can see here the diffusion term. And then this nonlinear part is written as a series of J's, and each one of these can be mapped to a different type of current. So this would be the sodium current, the potassium, and the calcium currents. And there's no term that is a conductance of the channels per se, so there are these constants tau, and what we did is assume that drugs that block this channel will lead to an increase in tau, and so forth for the others as well. So these are the drugs we had at our disposal: potassium blocker, calcium, sodium, and a blocker of gap junction, so it wouldn't affect any of these taus. And so this is the setup of the pins, once again, now with even more terms to account for the three variables. And if you look at synthetic data, it reproduces the time course for each of the variables very well, although it only has data for you. And this is the typical training setup to estimate one of the tau parameters. So it starts off with some excursions and then ends up converging to the ground truth. So this is the data. So for each drug, we have a pair in the same cell, the setup of data with no drugs and data with a drug, and then the pins reproduction of the action potential overlaying it. And so what we found was that our model was able to meet our expectations. So for drugs that we expect to affect potassium channels did lead to a significant increase in the corresponding tau, and so forth for the other drugs as well. So this is a setup where we spent a lot of time cleaning the data, and we were able to have good model agreement with it. In other cases, this is a little bit more difficult. So if you have more than 3 equations, it gets quite complicated to run the pins, in our hands at least, in inverse mode. It works fine in forward mode, where you're just solving the equations, but it has some difficulty estimating the parameters. So we found a workaround that by using the forward mode inside an optimization routine. So see if you can follow what I'm trying to explain. Say you have a set of ODEs here for which you are using your pins to approximate. You increase the dimensionality of your system on purpose by adding the parameter as another dimension. So now what you have is the pins that approximates the system for, in this case, for all time and for all the values of the parameter in a given interval that you've set. And as I said before, there's no discretization, so it really is in any point of this domain. So this is ideal once you've trained it. It's ideal if you want to do several passes to estimate this function, such as in an optimization routine. Also, if you want to use gradients in your optimization, they come for free from the way the network is configured. So that's what we've been doing. And the data I'm going to show is, oh, sorry, I'm going rogue, is from the brain. It's an fMRI data set. Someone goes in the MRI scanner and are shown nothing, or this checkered pattern intermittently, and then you keep track of the signals of the MRI intensity in the visual cortex that's processing the signals, and it goes on this kind of excursions. And this is a fairly complicated process, neuronal activation, coupling with hemodynamics, and then with the MRI process. And the most parsimonious model that we found to describe this has 6 coupled ODEs. So pins in inverse mode performs relatively poorly here. But this setup performs very well, and we're able to estimate several physiological parameters related to this physiology from this. So this was the kind of the complicated model problem. And then I'll also show you some data where we have very, very noisy data, and we're trying to run pins on it. And we've made some adjustments there. So this is also brain data. It's brain perfusion data in babies, so as noisy as it gets. And so the setup is that we're looking at the images of the brain in kind of this orientation. From a baby, you can see it here, the orange bit, and you've introduced something in the bloodstream, the arterial bloodstream, that makes the blood bright in MRI images. So first you see, this is a movie of images quite at different time points following this bolus injection. So first you only see signal in the arteries, then in gray matter, and at the end more towards veins. And if you want to estimate perfusion, that is the amount of blood that reaches each voxel in the brain per unit time, then you solve an ODE for each voxel. It's the same ODE, but you solve it separately because your perfusion is likely to be different, and so is the arrival time of this labeled blood bolus. And the other parameter in the model is global, is the T1 of blood. So this is the typical solution for the ODE, and this is a typical set of data. You can see noisy was an understatement of how bad it is. So we made some modifications to cope with this. We introduced a relative data agreement error where the noisier points have a lower weight compared to the ones that have less error, and this helped. And we also included a multi-branch pin. So instead of looking at each voxel separately and solving different ODEs for each one of them, we solve them all together. And then we come up with individual parameters for the local variables and the global parameter, taking all the data into account. So if you compare this with least squares or the vanilla pins, it performs quite a lot better. This is the relative error, except for arrival time, for which it's quite sensitive. So this is kind of what I wanted to talk to you about today. I hope I've convinced you. Pins are an interesting tool to combine mathematical models and data learning, that we can use it to look at cardiac electrophysiology in particular, and to estimate physiological parameters, also for experimental data, and I showed the example of the antiarrhythmic drugs for that. And then there are a couple of problems still that need adjustments of these typical pin setup for these aspects in particular. And this can get a little bit complicated because different people have different recipes of how to adjust pins. So if you want to dig in, but you're feeling that you want someone to really explain things to you slowly, and you're coming to Mikai in Morocco in October, then we have a tutorial there about pins, and I'd like to invite you all to join us there and learn more about it. And thanks very much for your attention. And yeah, if there are any questions, I'll be happy to take them.
Thank you, Marta, for very instructive talk. Any questions?
Very interesting talk, Dr. Kusma from Software in Glasgow. So a limitation of pins is that by optimizing the objective function, you get a point estimate of the parameters. How do you go about the uncertainty quantification? I realize that you provided some uncertainty intervals. Is that you basically do some sort of getting second order derivative matrix at the optimum, or is there any way you can generalize that to a more Bayesian approach where you then sample rather than optimize the objective function? Or will it be computationally expensive?
No, it wouldn't be. There is a setup for a Bayesian estimate of uncertainty, which is part of this zoo that I was describing. Everyone has a slightly different implementation of pins that is hard to navigate. But we haven't tried that. It's very computationally expensive, as we hinted. So my uncertainty just comes from the uncertainty in the neural network itself. It's just running the same neural network with different initialization seeds or different initialization values for the parameters that we're trying to estimate, and then just doing statistics on the different estimates. So it's much more basic than what you were suggesting.
Do you think that could be extended to go from a physics-informed neural network to a physics-informed Gaussian process, where you would get the uncertainty quantification for free, basically?
Yes, there is a setup for this, and I can pass on the reference for that if you're interested, but I haven't used it directly. For this kind of setup, but something more general is creating a type of pins called DeepONet that solves the entire setup of equations instead of being for point data like I was showing you here, for Gaussian models of the inputs, and then gives you the corresponding outputs with some uncertainty put in. So that's also available, but again, I haven't used that directly. So there's a lot of that there if you're interested. This is a relatively simple application of those techniques.
Any other questions?
For the optimization you described, where the P was optimized outside training of the network.
Yes.
So was that a kind of outer optimization, then inside the loop you would train the network, retrain the network?
Oh, sorry, I explained that super poorly. No, you train the network for a while. And I gave an example which was a two-dimensional example, but it's easy and it's not too complicated to extend it. So you can have your inputs, your time and your space, and then add the dimensions for the parameters, and you trained it for the domain of the parameter space that you're interested. And then once you've done that, you have an algebraic, what's kind of a solver for the equations for those parameters, so you can put it in an optimization loop afterwards, or using whatever optimizer you want. And then if you want to use the gradients for the optimization as well, you've also calculated them already.
I didn't quite, and the limit was backpropagation is slow or something to include it as if you were one parameter in the net.
So it's an empirical finding that it performs poorly. I think it's because there may be it's kind of complicated optimization setup where you're trying to find your function that is a solution to your set of equations, and you're trying to find the parameter as well. And in the beginning, there's no way to figure out which way to go about it. So that's why typically when you're doing an inverse mode, sometimes the network has a complete excursions of the wrong parameter value because it's still trying to find the shape of the function, so to speak, and neglects the parameter. Sorry, this is the vaguest hand-waving explanation ever, and neglects the optimization of the parameter as a consequence. And this is sometimes it never gives proper values, and it gets worse as you get more and more equations into the model. Again, this is all super empirical. If anyone knows a good way of making this quantitative, I'd love to hear that. But the way we bypassed that was to use the pins in forward mode and then put it in a classic optimizer for that.
Any other questions? I have one. The nice study on the rhythmic drug study that you did, was that just one cycle length, or do you did it for different cycle lengths?
We had data for different cycle lengths, but this is only using one cycle length, yeah. I mean, because it was very incomplete, so we had some with drugs, but not with the equivalent baseline and so forth, so we couldn't expand that. But that would be something really interesting to do. I was hoping you had a better idea to model conductance than the hack
Use a better
that
model?
than the hack that we used. But it seemed to match what we were expecting the drugs to do, which was nice, just as a proof of concept.
It was a nice fit, yeah. All right.
To follow up on Gary's question, because that's really a fundamental question, pins, isn't it? So do we want to optimize the parameters, or do we want to add them as an extra dimension? So you're saying that there's more your general experience, that adding them as an extra dimension is better, or what it varies application specific?
So from my experience, it's the inverse mode. When I introduced pins, I said forward mode, inverse mode. The inverse mode is super fiddly. And if you go through the pins papers, most of them work in forward mode and don't mention the inverse mode problems very much. And the way we found to work around that was to use them as a forward solver, and the idea of creating this extra dimensionality, which is related to the parameter space. I mean, it really is our idea. I haven't seen it elsewhere, but if you have and know more about it, let me know as well. But that's just a hack to make use of the pins expressivity, if you want to call it that, without being stuck in that inverse.
Yeah, I think that's important. Whenever I came on pins, I was wondering why people were not doing it the way you said, just suggested. And that most papers optimize the parameters basically along with the other neural network parameters. Whereas what you suggest is sort of the more natural route, I would say, than building an emulator.
Yes, so I think there are a lot of parameters that just work in silico setups, and then they bypass all the other parameters that I was pointing out. And then there are a lot of papers that just run it in forward mode as an equation solver, because the other is very tricky. I'd say that's the most common state.
All right.
Thank you.
Thank you, Marta. Thanks, Marta.